In 1991, a remote-controlled robot was sent into the darkest, most dangerous place on Earth, the sarcophagus of reactor number 4 at the Chernobyl nuclear power plant. 5 years had passed since the catastrophic meltdown, and the radiation levels inside were still so intense that they would kill a human being in minutes. The scientists monitoring the robot expected to find a biological wasteland. They assumed that in such a hellish environment, bathed in high-energy gamma rays, life would be impossible. But as the robot's grainy camera panned across the ruined, radioactive walls of the reactor core, they saw something that should NT have been there. Black mold. It was clinging to the wreckage, creeping up the walls. And it wasn't just surviving, it was thriving. Even more unsettling, the fungi appeared to be growing toward the sources of the most intense radiation, exhibiting a behavior that looked remarkably like a plant growing toward sunlight. Scientists called this radiotropism. This accidental discovery in the ruins of Ukraine kicked off a scientific detective story that has led us from the exclusion zone of Chernobyl to the pristine laboratories of NASA, and soon, potentially, to the surface of Mars. What we found is that these fungi possess a superpower called radiosynthesis. They can capture ionizing radiation and convert it into chemical energy. And this biological anomaly might just be the key to solving the biggest hurdle in deep space exploration, keeping our astronauts alive. To understand why a patch of black mold is so revolutionary, we have to look at the invisible enemy waiting for us beyond Earth's magnetic field. Right now, on the International Space Station, astronauts are still largely protected by the Earth's magnetosphere. But a trip to Mars is a different beast entirely. Once you leave that magnetic bubble, you are exposed to the full fury of the cosmos. The primary threat comes from galactic cosmic rays, or GCRs. These aren't just rays of light. They are heavy atomic nuclei, stripped of their electrons and accelerated to nearly the speed of light by supernova explosions in distant galaxies. Think of them as subatomic bullets. Here is the engineering nightmare. If you try to block a galactic cosmic ray with a traditional metal shield, like the aluminum hull of a spacecraft, you often make the problem worse. When a high-energy GCR hits a metal atom, it shatters the nucleus, creating a spray of secondary radiation, neutrons, and lighter particles that scatter like shrapnel inside the ship. This secondary spray can be even more damaging to human tissue than the original ray. To stop GCRs effectively without this spray, you need materials rich in hydrogen, like water or polyethylene, or immense mass, like lead. But mass is the enemy of rocket science. Every kilogram we launch to Mars costs thousands of dollars in fuel. We call this the mass penalty. Building a lead-lined shelter on Mars is logistically impossible with our current propulsion technology. We simply cannot launch a pre-built, heavy shielded habitat from Earth. This is where the fungi come in. The species found at Chernobyl, specifically Cladosporium sphaerospermum, share a defining characteristic. They are melanized. They are rich in melanin, the exact same pigment that gives human skin, hair, and eyes their color. For a long time, biologists thought melanin in fungi was just a stress shield, protecting the cells from UV light or temperature swings. But researchers at the Albert Einstein College of Medicine proposed a radical hypothesis. What if melanin is to radiation what chlorophyll is to light? The science behind this is fascinatingly complex. It involves a process of electronic alteration. Melanin is a complex, disordered polymer. When a high-energy photon strikes a melanin molecule, it alters the electronic structure of the pigment. It excites the electrons. In laboratory experiments, irradiated melanin showed a 4-fold increase in its ability to transfer electrons compared to normal melanin. The fungi use this high-energy electron state to drive a reduction-oxidation reaction, specifically reducing NAD+ to NADH. If you remember your high school biology, NADH is a primary carrier of energy in cellular metabolism. The cell uses it to produce ATP, the universal fuel of life. Essentially, this fungus eats radiation. In tests, Cladosporium grew significantly faster when exposed to radiation 500 times higher than background levels. This moved from theory to practice in 2018 and 2019, when NASA, in conjunction with student researchers, sent samples of this fungus to the International Space Station. They monitored its growth over 30 days. The results were staggering. A lawn of fungus, just 1.7 millimeters thick, absorbed about 2.1% of the incoming radiation. That might sound small, but the attenuation follows linear physics. Researchers calculated that a layer of this fungus about 21 centimeters thick, roughly 8 inches, would be sufficient to negate the annual dose equivalent of radiation on the surface of Mars. This changes the entire architecture of space travel. Dr. Lynn Rothschild, a senior scientist at NASA Ames, describes our current approach as the turtle strategy. We carry our house on our back. We build a hard shell on Earth and drag it through the void. It is safe, but it is incredibly inefficient. The new paradigm is the bird strategy. A bird carries nothing but itself and its DNA, and it builds a nest using resources found at the destination. This philosophy has given birth to a new field called mycotecture, architecture made of fungus. The vision for a Mars habitat under this model is not a tin can, but a living organism. The Mycotecture Off-Planet Project proposes a habitat design that acts like a three-tiered biological shell. The outermost layer is ice. We know there is water ice on Mars. We harvest it and fill the outer shell. This protects the inner layers from the vacuum and extreme temperature swings. The second layer is the farm. Fungi are heterotrophs. They cannot make their own food from sunlight. They need sugar, so directly under the ice, we place a layer of cyanobacteria, blue-green algae. Light passes through the ice. The algae perform photosynthesis using Martian CO2 and sunlight, and they produce oxygen for the astronauts and sugar for the fungi. The third, innermost layer is the mycelium. This is the vegetative part of the fungus, the root structure. We feed it the sugar, and it grows. As it expands, it binds together a substrate. This could be a lightweight mesh we brought from Earth, or even processed Martian soil. It acts as a biological glue, forming a dense, tangled mat that is stronger than concrete by weight, fire-resistant, and an excellent insulator. And, crucially, as it grows, it deposits that melanin into its cell walls, creating a black, radiation-absorbing inner lining for the astronauts. Think about the logistics of this. You don't launch a house. You launch a compact, folded plastic shell that weighs very little. Inside, you have pockets of dormant spores and freeze-dried algae. You land on Mars, you inflate the structure, and you add water harvested from the Martian surface. This wakes up the biology. Over a period of days or weeks, the habitat literally grows itself. The algae bloom, the fungi expand, and the walls solidify. Once the structure is fully grown, you have a choice. You can heat treat it, basically bake the building, to kill the fungus and leave behind a strong, inert biological brick, or you can keep it alive. If you keep it alive, you gain advantages that no metal habitat can offer. Self-regeneration. If a micrometeoroid punctures a metal hull, you have to patch it. If it punctures a fungal wall, and the organism is still alive, it can grow back and seal the hole. It is a self-healing home. But the potential of this technology goes beyond just walls. We are talking about synthetic biology. We can edit the DNA of these organisms to make the habitat a multifunctional biological factory. Imagine the walls are not just shielding you, but keeping you healthy. Researchers are already studying how fungi metabolize drugs in space. If an astronaut develops a bacterial infection or suffers from bone density loss on a 3-year mission, they can't wait for a resupply from Earth. In a mycotecture habitat, we could genetically trigger the living walls to secrete antibiotics or osteoporosis medication. You would literally scrape your medicine off the wall. We can also use them for biomining, Fungi are excellent at extracting minerals from rock. Radiotrophic fungi could be engineered to extract rare earth metals from Martian regolith during the growth phase. They can filter wastewater, breaking down urea and toxins into usable nutrients. And, potentially, the biomass itself could be processed into protein-rich food. The house becomes the farm, the pharmacy, and the water treatment plant. Interestingly, this futuristic technology is already solving problems right here on Earth. The principles of mycotecture are being applied by architects like Christopher Maurer of Red House Studio to humanitarian crises. In Namibia, for example, there is an issue with an invasive bush species that destroys groundwater supplies, alongside a dire housing shortage. The myco-HAB project harvests this invasive bush, grinds it up, and uses it as a substrate to grow oyster mushrooms. The mushrooms are sold for food, providing revenue and nutrition. The leftover root material is pressed into bricks and baked. These mushroom bricks are stronger than concrete. They are carbon negative, and they provide excellent insulation against the African heat. It is a closed-loop economy, where waste is just a resource in the wrong place. That is the exact mindset we need for Mars. Of course, we cannot talk about bringing alien-like fungi to Mars without addressing the risks. The biggest concern is planetary protection. If we bring a hyper-resilient radiation-eating fungus to Mars, and it escapes the habitat, it could theoretically colonize the planet. Mars has high radiation and subsurface ice, the exact conditions this cladosporium loves. If we later find life on Mars, how would we know if it's native or if it's just the descendants of the mold we brought with us? Worse, if there is native life, our aggressive fungus could wipe it out. NASA takes this incredibly seriously. The solution involves genetic firewalls. Scientists are working on kill switches in the fungal DNA. For instance, the fungus could be engineered to rely on a specific amino acid that does not exist in nature and must be supplied artificially by the astronauts. If the fungus escapes the habitat and stops receiving that supplement, it simply dies. It starves. Alternatively, the standard procedure might be to grow the habitat and then sterilize it completely before humans arrive, sacrificing the self-healing ability for ecological safety. There are also operational challenges. The simple phrase "just add water" hides a massive engineering hurdle. Water is heavy. To save launch mass, the water must be harvested on Mars using in-situ resource utilization. This means robots have to land, drill, extract water, and inflate the habitat months or even years before the astronauts ever leave Earth. The timing and reliability of those robots become the linchpin of the whole mission. But despite these challenges, the shift in perspective is profound. For the entire history of human spaceflight, we have fought against the environment. We have built thick metal walls to shut out the cold, the vacuum, and the radiation. We have viewed space as a hostile void to be conquered with brute force engineering. Radiosynthesis and mycotecture suggest a different path: cooperation with nature. By harnessing an organism that evolved in the radioactive ruins of our own mistakes at Chernobyl, we may find the key to our future among the stars. We are moving toward a future where our spacecraft are not built, but grown, where our shields are not dead metal, but living skin, and where the harsh radiation of the cosmos is not just a threat, but a fuel source. The first house on Mars might not be a gleaming dome of steel and glass. It might be a mushroom, dark, quiet, and quietly eating the cosmic rays that would otherwise kill us.